Hi everyone, today I will be using the Japanese art of suminagashi to show the intricate and beautiful development of something that you're relying on this very moment while you sit at home watching this YouTube video, and that is your respiratory system. I thought this medium would be perfect for this topic as it uses surfactant, which lowers the surface tension of water in order to enable the ink to float on the water surface, and as you will see later on in this video, the same substance is crucial for the function of your lungs. The organs of the respiratory system begin development at week 4 from the laryngeotracheal groove of the primordial pharynx that develops right below the four pouches and will become the tracheobronchial tree that will develop into the pulmonary epithelium and glands of the larynx, trachea, and bronchi from the endoderm. The intraembryonic splanchnic mesoderm produces the connective tissue, cartilage, and smooth muscle in these structures, as well as the blood vessels of the lung and visceral pleura. By the end of the 4th week, the laryngeotracheal groove elongates ventrally into the laryngeotracheal diverticulum that separates from the pharynx but still remains connected through the primordial laryngeal inlet. The laryngeotracheal diverticulum eventually grows to become the bulb-shaped respiratory bud with longitudinal tracheoesophageal folds fusing to create the tracheoesophageal septum at the end of the 5th week to divide into the laryngeotracheal tube and esophagus. Now, sometimes this can go haywire and something called a tracheoesophageal fistula can form instead. While there are many versions of this, the one that occurs in 85% of cases results in esophageal atrasia, an abnormal closure of the esophagus that causes amniotic fluid, milk, or any other substance that would normally be swallowed to be regurgitated upon entering the trachea instead. As you can imagine, this would bring a whole new meaning to the phrase inhaling one's food. The respiratory bud at the end of the laryngeotracheal tube continues to develop into primary bronchial buds that subdivide into secondary, then tertiary bronchial buds before further differentiating into various bronchioles of the future lungs. The lungs themselves are surrounded by pleura described later that contain the pleural cavity, which forms from the intraembryonic xylem, aka spaces in the lateral mesoderm. As the embryo undergoes lateral folding, the space that was originally open to the extraembryonic xylem is cut off from it by the end of the 4th week, so that the intraembryonic xylem is surrounded by visceral mesoderm and parietal mesoderm that derive from the somatic mesoderm and splanchnic mesoderm respectively. From a bird's eye view, the intraembryonic xylem is seen to actually be the shape of a horseshoe and is roughly divided into 3 parts. The pericardial xylem next to the developing heart that connects the 2 lateral pericardio-peritoneal canals that flank the tracheoesophageal tube on both sides by the 5th week and ends in the peritoneal cavity. During the 5th week, the growth of the bronchial buds causes the visceral pleura to touch the parietal pleura, even pushing into the somatic mesoderm by week 6. Now let's look at growth at a more detailed level through the 4 stages of lung maturation. First is the pseudoglandular stage that occurs between 5 to 17 weeks and is named such as the growth of the terminal bronchioles at this stage resembles a gland. The second is the canalicular stage at between 16 to 25 weeks, which is characterized by the formation of respiratory bronchioles from budding at the terminal bronchioles and a greater number of capillaries. Near the lateral portion of this stage, respiration can become possible if some primordial alveoli are formed from terminal sacs developing on some respiratory bronchioles. Next is the terminal sac stage that occurs between week 24 to birth. Again, as the name suggests, more terminal sacs form and we see the differentiation of cells into 2 types. Pneumocytes and secretory alveolar cells that are type 1 and type 2 respectively. Here the capillaries move adjacent to terminal sac. Finally, we have the alveolar stage that continues for up to 8 years from the latter fetal period and involves the maturation of terminal sacs into fully functioning alveoli that can perform gas exchange due to a thin membrane between the alveoli and capillaries. Here we also see more type 2 cells capable of secreting surfactant, that special substance made of phospholipids and proteins that lines the alveolar lumen to decrease surface tension while also increasing lung compliance, which is the change in lung volume produced by a unit change in transpulmonary pressure. Insufficient surfactant can result in the collapse of alveoli, resulting in greater effort being required to breathe and can be potentially fatal, and has even been linked to respiratory distress syndrome in infants born before the 28th week of pregnancy. Luckily, this has prompted research into animal-derived and synthetic surfactant replacement therapy, and steroids given to the mother to mature the baby's lungs is another treatment option if the problem is discovered prior to birth. Fun fact, recent studies have also found that surfactant plays a role in host immunity against pulmonary infections. Unfortunately, we're out of time, but I just want to give a quick nod to the diaphragm, which separates the thorax, which contains the respiratory system, from the abdomen. Anyways, next time you're doing a pandemic-safe activity like hiking, riding a bike, taking a stroll, watching Netflix, or trying to make your very own human embryology-inspired art, remember, even though simply breathing might not seem like much, there were numerous steps and checkpoints that had to be reached for this to be possible. Also, for your chance to win one of my very abstract prints made in this video, comment hashtag ANA301 with another embryology art suggestion. Thank you for watching. Please like and subscribe.